Welcome to this explainer. Today, we're going to demystify the completely fascinating and sometimes a little complex world of enzyme kinetics and structure. We're going to take what might initially seem like dry, intimidating biochemical math and transform it, because in reality, this math is the foundation of how modern medicine targets the very engines of human life. By the end of this, you'll see exactly how invisible molecular folds, slight temperature fluctuations, and some really clever equations literally dictate health, disease, and the specific medications that save lives every single day. Let's dive in. It's a profound mystery, isn't it? Kind of wild if you stop and really think about it. You swallow a tiny pill, and somehow it knows exactly how to target a rogue bacteria or lower your cholesterol without messing up anything else. Well, the answer to this mystery lies entirely in understanding biological catalysts, our enzymes, at a microscopic level. You see, almost every drug in existence is essentially a precisely engineered wrench being thrown into a very specific biological engine. But it's not just any wrench. The precision required to do this without breaking the rest of the machine is absolutely staggering. We're talking about structures so incredibly tiny that a single misplaced atom in that drug can render it completely useless, or even worse, incredibly dangerous. The sheer scale of this microscopic targeting, that's what makes biochemistry the true foundation of pharmacology. All right, here is our roadmap for today. First, the architecture of biological catalysts. Second, the math of metabolism. Third, stopping the reaction, inhibition. Fourth, regulating the body's chaos. And finally, number 5, enzymes in modern medicine. Okay, let's start with section 1, architecture of biological catalysts. So to truly grasp how metabolism works, we have to start at the absolute smallest scale, the physical architecture itself. And the evolution of how we understand this is just brilliant. For a long time, science viewed enzymes through what we called the lock and key hypothesis. We basically thought of the enzyme as this rigid, static lock, and the target molecule, the substrate, as a perfectly shaped key. But the reality, it's far more elegant than that. Today, we use the induced fit model. We moved away from the idea of rigid locks, and instead, we recognize that enzymes are these flexible, dynamic proteins. Their active site, which is this tiny 3D pocket, actually molds and shifts the moment the substrate binds to it. This conformational change literally stresses and bends the chemical bonds of the target molecule, which vastly lowers the activation energy needed for a reaction to even happen. Imagine you're trying to snap a really thick stick in half over your knee. If you just press on it, it's tough, right? But if an enzyme wraps around it and pre-bends it for you, it snaps with almost no effort at all. That dynamic embrace is what makes life possible at the relatively low temperatures of the human body. Moving right along to section 2, the math of metabolism. Okay, so how fast do these biological reactions actually happen? Well, we translate that physical 3D shape into mathematical speed using the Michaelis-Menten framework. And we'll start with Vmax. Think of a biological factory. Vmax is the absolute maximum production speed possible when every single enzyme worker on the assembly line is fully occupied or saturated with raw materials. You literally cannot make the reaction go any faster no matter how much more raw material you dump into the factory. Why? Because the workers are already operating at their absolute theoretical limit. When we graph this out, once that factory is fully saturated, the curve completely flattens out into a horizontal line. It perfectly visualizes the exact moment where the enzyme itself becomes the ultimate bottleneck for the reaction. Next up, we have Km, otherwise known as the Michaelis constant. This is the exact amount of substrate required to get our factory running at exactly half of its maximum speed. So think about it. Km acts as an inverse measure of affinity. Let's go back to our factory. A remarkably low Km is like having a highly magnetic worker on the line. They can grab passing raw materials even if there are barely any around, hitting half speed almost instantly. A high Km, on the other hand, that means the worker is essentially wearing thick, clumsy gloves. You have to literally bury them in raw materials just to get them to work at half capacity. So the takeaway here is simple. The lower the Km, the tighter, more eager the biochemical embrace. But what if we want to zoom all the way in on just one individual worker in that factory? To measure the pure catalytic efficiency of a single isolated enzyme, we look at the turnover number, or kcat. This tells us exactly how many molecules one specific enzyme can process in a given unit of time when it's going as fast as it possibly can. But honestly, it gets even better. By looking at the ratio of kcat over km, biochemists can perfectly measure overall catalytic efficiency. This ratio represents the perfect balance between how unbelievably fast an enzyme works, its kcat, and how easily it grabs its target, its km. Enzymes that maximize this ratio, they're crowned the most efficient catalysts in the known universe. They work so flawlessly that literally the only thing slowing them down is how fast molecules can diffuse through water to reach them. It's mind-blowing. Now, on a standard graph, an enzyme speed creates this sweeping curved line that approaches Vmax like an asymptote. But actually, scratch that, because determining a precise maximum velocity from a curved line is notoriously difficult. So biochemists mathematically linearize it into this double reciprocal graph called the Lineweaver-Burk plot. By plotting 1 over velocity against 1 over substrate, the data forms a perfectly straight line. Now, because it's a double reciprocal plot, everything is flipped, which, yeah, can be slightly counterintuitive at first glance. A larger Vmax actually means a smaller y-intercept, pushing the line closer to 0. But breaking down this slightly weird math reveals a secret code. It gives us the exact hard numbers we need to map out precisely how different conditions, and especially drugs, will alter an enzyme's core function. Which brings us to section 3, stopping the reaction, or inhibition. So manipulating these specific variables is exactly how life-saving pharmacotherapy works. Let's say a patient has high cholesterol, we give them a statin. Statins are competitive inhibitors. They literally compete with the natural substrate to get into the enzyme's active site. Because of this direct competition, the enzyme's overall affinity drops, so Km increases. But if you flood the system with enough natural substrate, you can overwhelm the drug and still eventually reach the normal Vmax. Non-competitive inhibitors, though, they're a bit darker, like heavy metal poisoning. They bind to a completely different spot on the enzyme, an allosteric site, and warp the enzyme's entire shape. No matter how much natural substrate you add, the enzyme is broken. Vmax permanently decreases. Finally, there are rare uncompetitive inhibitors. These are super unique because they only bind to the enzyme after the substrate is already inside, effectively trapping it there. This locks the complex together, forcing both Vmax and Km to decrease at the exact same time. All right, section 4, regulating the body's chaos. Long before we ever introduce any artificial drugs or poisons, the human body itself has to tightly control these enzymatic engines just to prevent total metabolic chaos. 37 degrees Celsius. This specific number represents the absolute optimal temperature peak for human enzymes to function, and it is a fascinating evolutionary balancing act. Heat increases kinetic energy, right? Making molecules bump into each other faster, which speeds up our biological factory. But if it gets too hot, the machine physically melts. 37 degrees is that precise evolutionary sweet spot between maximum metabolic speed and vital structural stability. If a patient deviates too far from this optimal temperature, say during a really severe, prolonged fever, or strays from an enzyme's precise pH requirement, the results are catastrophic. The critical non-covalent interactions and ionic bonds that hold that 3D shape together just begin to unravel. The protein denatures. And this isn't just a temporary pause. Denaturing is a permanent, catastrophic unwinding of the protein. The engine breaks down completely. It's exactly why enzymes in your highly acidic stomach, like pepsin, function perfectly at a pH of 2, but would denature instantly and become entirely useless if they moved into your pH-neutral small intestine. The body also has to protect itself from its own creations. To survive producing its own destructive proteins, the body relies on zymogens, which are basically inactive precursors. Take the digestive enzyme trypsin. If the pancreas produced active trypsin right away, the enzyme would literally digest the organ that made it, causing severe, life-threatening pancreatitis. Definitely not what we want. Instead, the body creates an inactive, perfectly safe version called trypsinogen. It travels safely down to the duodenum, where it undergoes a very precise, irreversible cut called proteolytic cleavage. Suddenly, it wakes up, becomes active trypsin, and starts digesting your food instead of your internal organs. Talk about a crucial safety mechanism. And that brings us to our final stop, Section 5, Enzymes in Modern Medicine. So, what's the clinical payoff here? How do we use all this in a hospital setting? Primarily through diagnostics. When cellular structures are damaged or die, a process called necrosis, their outer membranes literally break open.
Their highly specific intracellular enzymes leak out of the broken cells and wash straight into the bloodstream. By measuring these serum levels, doctors get highly accurate liquid biopsies. For example, if a patient's blood is suddenly flooded with creatine kinase, specifically the CKMB isoenzyme, it instantly reveals that a heart attack is occurring. High levels of ALT point directly to liver damage, like hepatitis. And if amylase or lipase are detected at extreme levels, it points us right back to the pancreas, diagnosing acute pancreatitis. The simple math of tracking these enzymes leaking into the blood is absolutely the cornerstone of modern clinical diagnosis. Furthermore, our deep structural knowledge dictates how we build the world's most powerful drugs. You see, an enzyme doesn't alter the overall free energy of a reaction, it accelerates it by stabilizing the most highly energetic, unstable midpoint of the reaction, known as the transition state. Think of the transition state as the exact moment a heavy bow is pulled back to its absolute maximum tension right before firing. It's incredibly unstable, right? Well, the enzyme is built to perfectly cup and hold that fully drawn bow. If we design a drug that looks exactly like a permanently drawn bow, a transition state analog, the enzyme grabs it, but because the drug is a fake, the enzyme gets hopelessly stuck holding it. That is why HIV protease inhibitors are so devastatingly effective against viral replication, and why penicillin can effortlessly halt bacterial cell wall synthesis. The enzyme grabs the drug, and it literally never lets go.
So, what does this all mean? Well, every concept we've explored today shows us that health is really just a delicate, intricate balance of molecular geometry and kinetics. Since genetic mutations can easily build toxic internal roadblocks by even slightly altering an enzyme's Km or Vmax, understanding these mechanics is absolutely vital for the future of personalized medicine. We've journeyed all the way from invisible, microscopic folds and mathematical graphs right up to the very macroscopic humans walking around a hospital ward. So I leave you with this to wonder. As science gets better at analyzing our DNA, what complex biochemical mysteries, unique affinities, and potential metabolic blocks are waiting to be solved within your very own cells? Thank you so much for exploring the engines of life with me today. Catch you next time.